done was give you an introduction to the concept of breeding self-pollinated crops using either a bulk selection method or a pure line selection method. And I was going to talk about in early generations or early breeding programs for self-pollinated crops. You could do that very readily because there was enough variation, useful variation, present in land races. So what we'll do is we'll pick up today to point out, and then we'll go through the stages, as the variation available from native land races sort of decreased. I mean, there's still a lot of variation in land races, but today they're used primarily to go look for genes for specific traits for abiotic stress tolerance or disease or insect resistance, or in some cases quality type traits. They're not used as source populations that much because even though all of this genetic variation in land races has some useful traits, as you might guess, 90% or more of the traits in those land races aren't very useful. They either replicate what we already have in domesticated varieties or they're detrimental. And so today in most self-pollinated crop breeding programs, we go through a stage of hybridization to create genetic diversity, and then we get an F2 population that's diverse. And then breeding methods, we can either do a bulk method or any number of pure line methods. Just to backtrack just a little bit, as I said, originally the land races had lots of variability. They contain a mixture of several types of lines. So if you were breeding wheat 50 or 100 years ago, it was very easy and very useful to bring in a series of land races and just select individual lines, pure line cultivars out of those races, and you're often running with pure line varieties. They contain desirable alleles, but often many undesirable. Well, the extensive selection from those land races reduced the diversity and sort of pulled out the desirable alleles to put in our elite varieties. And so more recently, self-pollinated crop breeders are trying to use these varieties that already have pulled out desirable alleles from the land races and only go back to the land races if you need something specific. And so today, new and often more useful genetic variation is created via hybridization. What does that do? Hybridization creates diversity, and we've gone through this before. An F1 is completely heterozygous for all loci at which the two parents differed. As you self, you lose half of the heterozygosity, or you gain half again the homozygosity for each generation. The number of loci for which two parents differ affect how much variation you get in that F2. The number of alleles at each locus, if you have multiple alleles at loci and you have parents that contain, you don't just cross two individual plants, you often cross several plants for each parent type. And of course, linkage relationships affect the nature of recombinations you can get out of an F2. And just to give you a feel for the magnitudes and often how inept our plant breeding studies are at sort of measuring this variation, if the two parents that you pick differ at one locus, it's pretty easy. You can end up with two different types of F1 gametes and 3 F2 gametes and 4 different types of individuals in the F2 generation. Pretty simple Mendelian type genetics. It's easy to do. You drop down to if your two parents differ by 10 loci. Then you've got over 1,000 different kinds of gametes produced in the F1, almost 60,000 different kinds of gametes produced in the F2, and about 1 million different types of individuals can show up in that F2 generation. As I'll talk about later today, usually when we look at the F2 generations of our breeding populations to select plants, we try to get 1,000 to 2,000 individuals. So what are the chances that we identify the very best superior cultivar among those million segregating plants if we're only looking at 2,000 plants to begin with? So our chances of picking that really good genotype first time through a breeding program aren't very good. That's why plant breeding is a process that goes on year after year after year. We pick what we think might be the best few genotypes, then we cross those together to produce another population to try to ultimately capture all of the useful genetic variation that we can generate. So, all right, we'll talk about mass selection or just a bulking method, and we'll talk about all of the different pure line breeding methods. To sort of compare the two, you know, it's lumpers or splitters. If you like to lump things together in broad general categories, you'd make a great bulk method breeder. You just grow out the parents, cross them, produce an F1, produce an F2 generation, and select the seed from the best plants and bulk them together. And go F3 and bulk them together. Selecting the F4, bulk them together. And you just bulk everything together till you get down to about an F5 generation, which is about 94–95% homozygous, each individual plant. Then you plant the plants out in what we call space plots, so you can look at and evaluate each individual plant, and you select the best plants, and from each plant, you plant one progeny row from the seed from each selected plant. In the next generation, these progeny rows can then be replicated, and you can then start selecting, well, what are the best progeny rows to carry on to the next generation, and you eliminate a lot of the undesirable ones and only carry the best forward until finally you get a few that you can take to multiple location replicated trials. So ultimately, you can develop and release a variety. So basically, the bulk method says, I'm not going to waste my time during selection way up here. I'm going to create that variability in an F2, and then I'm just going to bulk things together until I get down to my fairly homozygous plant states, and then select individual plants. With the pure line or pedigree methods, you start in that F2 generation, you plant the individuals out in space plants, you select individual plants, and advance those plants to progeny rows. The next generation, you go in those rows and select individual plants, and each row in the next generation comes from one plant in the previous generation. Do the same thing through the F5 generation until finally at the F6 generation, you increase the seed of those progeny rows and you take them out to replicated yield trials to release the variety. We'll go through each process individually and individual steps, but basically in both of these processes, what we're trying to do is take a pure line variety, cross it to another pure line variety or something that's not so genetically uniform, and create genetic variation. But then in self-pollinated crop breeding, our products are pure line varieties, so we got to get back to homozygous varieties before we can really select and develop something worth releasing. Point out here, and I'll emphasize this several times, all of this selection up until we get to replicated trials is all based on phenotype. So that the individual breeder goes in and selects the plant that looks the best, the right maturity, the right plant height, the biggest head for a wheat plant or a sorghum plant, the most grain, the right grain color. So all phenotypic. Well, phenotypic selection is very good at selecting traits that are highly heritable. So it's very good for selecting these traits that aren't controlled by a lot of genes. Traits like yield and overall performance are controlled quantitatively, and so we really have to get down to some replicated yield trials to select and improve those quantitatively inherited traits. Why is that? When we get to quantitatively inherited traits versus relatively simple qualitative traits, what factor complicates our lives? Well, yeah, we'll talk about that next week. Inbreeding depression if you got to cross-pollinate it, but not just environmental variation. These traits are almost, you can almost say, simply inherited traits, so the environment doesn't play as big a role. These very complex quantitatively inherited traits, there's a genetic component and an environmental component, and until you can plant replicate plots to measure or estimate that environmental variability, you really can't do much quantitative selection for quantitatively improved genetics. All right, just go through these. Mass selection, it's great for inbreeding a segregating population. Basically, the seed used to grow each self-generation is a sample of seed that's just harvested in bulk. You just bulk F1 through F4. F5, you start selecting individual plants phenotypically. By the time you get to the F6, 7, 8, you start running progeny trials, replicated trials. And basically, it's a short-term, time and labor-saving, practical breeding procedure for managing segregating populations of inbreeding crops. You create the variation, select in bulk, select individual plants, select the best families for advance, test the experimental varieties, and release. Advantages: less record keeping. It's inexpensive, easy to handle, more crosses. It's the natural selection occurs in each generation. I mean, you only select the good-looking plants, so if a plant is in a bad location, or if a plant is late maturing or dwarf or has some undesirable characteristic, it's not selected for the bulk. You can maintain large numbers of genotypes. It works very well with unadapted germplasm, and you can carry it on for many, many years without a lot of expense or effort. Its disadvantages: your environment changes from season to season. So plants you select in bulk one season may not do so well. Their progeny may not do so well the next season. So as the environment changes, you're selecting slightly different genetic components from that population. You must grow that bulk seed in an area of adaptation. So you must do your selection every year in the environment where the farmer will ultimately grow the variety. Eliminates any chances for off-season nurseries or 2 or more generations a year. It's less efficient on highly heritable traits because we'll talk about in the pedigree selection, if you see a plant that's really bad, you can throw it out. If you're just grabbing and bulking seed, these are not, you can't look much at individual plants, because in these small grains, if you normally plant them at very high population densities, and you don't really get to see these undesirable phenotypes until you spread the plants out and give them a little space so you can look at individual plants. So you can really carry some undesirable types along. It's not useful in selecting plants at a disadvantage, dwarf types. And I mean, that might sound silly, but most of the wheat that we grow today are dwarf, and the sorghum varieties we grow today contain dwarf genes in order to give us short plants that will stand up and not lodge, fall over before harvest. The final genotypes may be able to withstand more environmental variability, but you'll never get the highest yielding genotypes from a bulk. Just won't do it. By definition, you're bulking the best yielding with some of the poorer yielding, and you're just not getting that high yielding one. And of course, if you try to do this with cross-pollinated species, inbreeding depression just drives you crazy. We'll talk about cross-pollinated species Monday. All right, the pedigree objective or method. The main objective, isolate superior, recombinant, homozygous lines. You start selecting in the F2 generation and select every generation onward. Response to early generation selection for quantitative traits may be low because of heterozygosity. Why? Why does heterozygosity interfere with your ability to select desirable alleles? It's kind of like a question on your exam. Why is additive genetic variance preferred over dominance? Because if you've got heterozygosity with dominant genes involved, you've got a bunch of undesirable recessive alleles that are hidden. And you select a lot of plants with a lot of heterozygous loci, well, the next generation, a quarter of the progeny from that plant are going to be homozygous recessive and not going to look very good. All right? So, to do pedigree method, and we can do this for either method, actually. This is sort of how you would make a cross in wheat. You grow 50 to 100 F1 plants. At least you grow enough plants to get, here we say 2,000 to 3,000 F2 individuals. Then you plant those F2 individuals. Select and harvest plants or spikes that seem to combine the desired characteristics from each parent. So we grow 2,000, 3,000 plants out, spaced out so we can walk through and look at each plant individually. Select individual plants that we like, and the next generation, each row are the progeny from one individual plant. We go through that for 3, 4, 5 generations until finally we select about 25 to 50 families that may be retained and increase to go into seed increase for replicated yield plots. Yeah.
I don't understand the use of the term families.
Term of?
Families.
A family is just a group of related plants, just like a family here is a group of related people. So that if you have, let me back up one. If we start out with that individual plant and we plant out the progeny, then those progeny are a family of related individuals from that plant. But it's a good point. So as we go here, we select 1, 2, 3 from that related family, and we get 3 more rows. Well, all of these rows are still part of that original family descending back from that original F2 selection. And so we treat each of these rows as a different family or subfamily until we get down to an F5 or F6 generation. We want to select about 25 to 50 of those to produce seed. In some cases, you could plant, if you harvest the seed from a wheat spikelet, you can get enough progeny to grow 1 or sometimes 2 rows. Not enough to grow 2-row replicated yield tests at 3 locations. So ultimately, you got to start bulking the seed from the different plants within your final version of the family to get enough seed to go to replicated testing. And of course, by the time you're bulking them at an F5 generation, they're 98% homozygous. So you're bulking, but you're bulking almost identical individuals. There's not a lot of variation within each of those families by the time you get to the F5 generation. So at the end, you grow the families in 1 plant rows and select designated seed sets and label them experimental lines. You grow the experimental lines together in a preliminary yield trial with standard varieties that are grown by farmers as checks. And you take those yield trials to 2 or 3 environments, and ultimately you increase the seed and distribute a new pure line cultivar.
How do you increase the seed? You just self-pollinated?
You put it in an isolation, so there's no chance, even though this is a self-pollinator, there's no chance of pollen blowing in. So you put it in an isolation, and you just let the plants self-pollinate to produce more seed. For a crop like wheat, where we have a fairly low outcrossing ratio, the isolation doesn't have to be very far. You get to sorghum, it has to be a little more isolated. By the time you get to rice, where you can get more frequent outcrossing, you have to have even greater isolation. So basically, the assumption is if you grow those plants a few meters away from other varieties you're increasing, you don't get enough outcrossings to the goal is to get a variety that's 98% pure.
So in the previous generation, when they're only wanting to get one plant, they're like putting a little bag over the head so that...
You can do that. And if you want to ensure all the way through that the plants you select are self-pollinated, you can just bag the heads and ensure. But again, with wheat, a lot of times in wheat, the pollination occurs before the flower opens. And so it's pretty much self-pollinated. So what are the advantages? It eliminates really disastrous, unpromising material at an early stage. You've got multi-year records, so you can follow not only the pedigree family background, but how those families performed each year, which was a different environment. And multiple families are maintained, but those multiple families all have maybe different gene combinations, but they have the same phenotype. So you can produce a pure line variety, but still a little bit of genetic diversity within that pure line variety. Genetic diversity that's there genotypically, but you really don't see much phenotypic variability. The disadvantages: labor, time, cost. So when you're using pedigree methods, you usually have to compromise between the number of hybrids, F2 generations that you work with, or the number of F2 plants you can grow out. So you could maybe start with 4 or 5 source populations and grow out the 2,000 to 3,000 F2 plants, or you could maybe go to 10 or 12 source populations and only grow out 1,000 F2 plants, but the size of your nursery is going to be limiting. And so the breeder has to make the decision, do I need more crosses and fewer F2 progeny for each cross, or fewer crosses and more F2 progeny to select from? So it's very dependent on the skill of the breeder. This concept that there's an art to plant breeding is not total fallacy. There are people who can actually look at these plants and can estimate very, very well from the size and the shape of the grain on the plant whether that's going to be a good high-yielding variety or not. And there are other people that can spend a lifetime looking and selecting plants and finding out that when they finally go through to replicated yield trials where you're actually quantitatively measuring yield, they just didn't make the right selections, you know. Unfortunately, in the public sector, those people continue, well, fortunately, in the public sector, those people continue to be very well-respected plant breeders, and they produce lots of articles, and they do lots of teaching. In the private sector, those people are out of a job very early because there's a whole new generation of younger breeders coming in, and the ones that really have that breeding art are the ones that are successful. All right, the other thing is it's really slow. You can only go one generation a year because, again, the selections that you make have to be in the environment where the farmer is going to grow your final variety. All right, so several people, Walt Fair at Iowa State with soybeans, I think initially started saying, boy, this is too slow. Plant breeding today is so fast-paced, I just can't go with one generation of selection a year. And this pure line selection is nice and has lots of advantages, but it's too slow. This bulk method is very good, but I take a lot of stuff forward that I don't really need. So they said, well, why can't we do a little modification so that we can do a single seed descent procedure whereby we basically advance generations by taking, you plant out your 3,000 F2 plants, and you take one seed from each that flowers in time. And you just advance it. The next generation, you take one seed from every plant that flowers in time. The next generation, you take every seed from a plant that flowers in time until you get down to that F6 generation. Then you plant all of those individual plants out and make your selections. So you can get to the F6 generation faster, and you can carry forward a maximum number of F2 plants, the genetics from those F2 plants, to a stage of homozygosity. So what it's based on is, in the pedigree method, there are really two phases. You develop those pure lines. You start with that F2 generation, and you select and develop pure lines by selecting in your target environment for 4 or 5 years. Then you select among pure lines to get the desired traits and put those pure lines in replicated trials. Well, the single seed descent says, well, let's just get, let's don't do any selection during those F3, 4, 5 generations. Let's just fast forward through those by taking single seed. Then when we get to that F6 generation, we're back to homozygous plants. We can start selecting varieties and not worry about all this preliminary stuff. And so the advantages are you can do 2 or more generations per year because you're not doing any selection. Your generations don't have to be grown in an environment where the variety will be grown, because you're saying, I'm just randomly bringing one seed from everything down. Very good for developing inbred pure lines for environments different from those in which the segregating populations work. I mean, if you're developing pure lines for hybrids, for instance, it works well. But it's also very efficient for pure lines varieties. So what you do, you cross-selected parents, produce F1 seed. You grow enough F1 plants to get 1,000 to 2,000 F2 plants. Then you grow your F2 plants under conditions that force early maturity. You grow them under conditions so they'll flower as fast as possible. Select one seed from each plant. Repeat through 5 generations. Get to homozygous F6 individual families and start doing your selection and release new varieties. So basically, take the F1, you go into the F2, and you just take single seed from single plants, all the way down till you get to the F6. Walt Fair, with his soybeans at Iowa State, takes the F2, takes it into a greenhouse, and he does F2, F3, F4, and sometimes F5 generation in the greenhouse. And the next summer, he's ready to plant out F6 individual plants for selection. So he can get from his F2 to his F6 generation in 1 year instead of 4. And the power in that speed, or that fast pace, is what really makes a successful breeding program. I mean, farmers can grow 1 generation a year, and farmers grow varieties that run into either years where they perform well or not, but farmers are always in need of new varieties. And if your breeding program is matching the farmer by growing 1 generation a year, year 1 the farmer says, I need something new, and you say, don't worry, 5 years from now I'll have it for you. The farmer says, wow, what am I going to do for the next 4 years? So if your breeding program should be running so that you've got material coming through so that you can stay 2 or 3 or 4 years ahead of the farmer's needs. Now, can you anticipate those needs? No. But, you have a lot of different lines down here to look at. And if all of a sudden you get a new disease or a change in environment or something that impacts the farmer's productivity, you say, wow, look at all of these things that look like my previously released varieties aren't doing well. But I have a few families here that look pretty good. And so I'm only 2 years away from being able to release something new rather than 5 or 6 years away. So its advantages: rapid generation, requires less time, space, resources in the early stages, so it can do a higher number of crosses. It's superior to the bulk or mass selection if the genotype is at a competitive disadvantage. I mean, it doesn't matter whether you're dealing with early, late, dwarfs, you're pushing the maturity on everything to get them to flower. The delayed selection eliminates any of the confusing effects of heterozygosity. By the time you get to an F6 generation of a self-pollinated crop, you don't have many heterozygous loci left at all. It's highly amenable to modifications, so it can be combined with any method. You could do single seed descent for 2 generations and then bulk everything. Or you could do single seed descents for 2 generations and then go to pure line selection. So it just gives you that flexibility. Disadvantages: you will carry some inferior material forward, but that doesn't matter because you're carrying so much more material forward that at the F6 you just forget about those inferior things. You really lose the advantage of natural selection, but then again, in most of our growing environments, that's not going to amount to a lot that you're losing. The biggest disadvantage is you need the facilities, some sort of controlled environment to get those generations through. Requires greenhouses or growth chambers or both. All right, so basically those are methods for breeding self-pollinated crops. You've got the 2 methods, the bulk or the pure line selection, where you actually go through a period of more or less selection, either bulking or selected for generations F3, 4, 5, and you wait till you get to the F6 generation to do that final phenotypic selection. Then you go to the advanced generations with replicated yield trials to really select for the traits with low heritability. Or you do the single seed descent where you say, those generations F2, 3, 4 don't mean a thing. Just rush through them, fast forward through them, get my F2s back to F6s that are homozygous, then I'll do the selection. And if I've thrown away some really good ones because they didn't produce a seed, well, too bad. If I've carried forward some really bad ones and my F6s look really ragged, that's okay, I'll throw away the bad ones. I don't worry about it either way because next year I'm going to have a lot more F6s coming through that my competitors in a pure line or bulk program aren't going to have for another 4 years. And so, you say sort of the speed is the power. All right, we'll talk about a couple of other, actually, at least we'll get through 2 other procedures that sort of apply to all types of crops. Backcrossing method is really, it's just a form of recurrent hybridization, and it's used to transfer a gene, preferably a single dominant gene, from a desired parent, or a gene from a parent that may not be that desirable, but has at least one simply inherited trait that's good, and move that gene into a desirable variety, a recurrent parent. So the trait should be simply inherited and easily phenotyped. The recurrent parent should be highly acceptable genotype, an existing commercial variety, hopefully. And the backcross donor will possess the trait you desire with not a lot of other undesirable stuff. So this is used a lot to transfer single genes for disease resistance, or a lot to transfer single genes for quality traits, single genes for any number of traits that you can phenotypically identify. As you backcross, it looks a lot like as you self to get the homozygosity. Each backcross generation brings you 50% back to the type of your recurrent parent, or takes you 50% farther away from the genotype of the donor parent, so that after about 4 backcross generations, you can get to about 97% back to your recurrent parent type. I pointed out in one of the early lectures, and we'll do it again when we talk about using molecular markers as tools. If you use molecular markers, you know, actually this backcross 1 generation is a normal population that segregates from plants that are anywhere from about 60% back to the recurrent parent type to plants that are about 85 or 87% back. So using molecular markers, you can actually do this in 1 or 2 generations. But without markers, you can't select those different genotypes. All you can select are the phenotypes, so it'll take you 4 generations. The process is you cross a disease-resistant cultivar by an adapted cultivar. You produce the F1 that's heterozygous for the resistance gene. You backcross it to your desirable cultivar that had no resistance gene. The backcross 1 carries 75% of the genes from that desirable cultivar. Backcross 2 gets 87.5, 93 3/4, 98.9, so that ultimately you get a population that essentially has most of the genes from your adapted parent plus the single gene that you've added. That single gene is still segregating, so then you go through a self-generation so you can select only the homozygous dominant plants carrying that resistance gene. So it has advantages, but it's not really a breeding procedure. It doesn't really create any new genetic variability. All it does is add, well, unless you consider that one single gene enough to give you genetic variation, all it does is it takes a pure line variety, and it can either be a pure line variety of a self-pollinated crop or an inbred parent of a cross-pollinated crop, and it adds 1 gene. All right, the double haploid method becoming more and more prominent in breeding programs. Basically, you generate haploid plants from the anthers of your F1 plants, and you never really go through an F2, 3, 4, 5, 6 generation. You take plants generated from pollen of your F1 plants, and you double the chromosome number such that you cross variety A by B, you bulk plot the F1, you culture the anthers, you develop haploid plants out of those anthers, you double the chromosome number. Sometimes with colchicine, and a lot of crops, they double spontaneously. Where? Wow, it's extracted from what plant? Do you know what plant it is? We can look it up. It's an extract from a plant. It's a compound that, is it crocus? Yeah, I think it comes out of a crocus plant. So, but Lisa Earle tells me that in a lot of our crops today that you just don't need, with rice for instance, you don't need colchicine. And so you get double haploids, and so your F3 generation then are individual pure line plants. I mean, each plant is genetically pure because you didn't have to go through all of that recombination through each sexual generation. You doubled, you take a haploid configuration and double it, and you get a plant that's homozygous for every locus. So it sort of speeds that process tremendously so that you can start doing your selections to get, and even though we say this is F3, 4, 5, actually what you get is the F1, the double haploids. You go 1, 2 generations of selection of the best plants among those double haploids, and you're up to an F5 generation. So very, very rapid way to get back to homozygosity. It's been used to some extent in self-pollinated crops, where breeders really make crosses and they want to get back to type in a hurry. It's beginning to be used in cross-pollinated crops like cassava, and we'll talk Monday about the problems you have when you self these crops to get them homozygous. Well, cassava, it seems like if you do a anthroculture and a double haploid, you can get a homozygous plant, and you can select homozygous plants that survive. And so you don't worry about inbreeding depression killing off most of the plants that you're selfing. All right, and multiline breeding. We'll talk about this a little bit later when we talk about breeding for disease and insect resistance. I mean, traditionally these self-pollinated crops were developed as pure lines, which meant if they were developed from a race of wheat in Turkey, they carried maybe one or more major genes for resistances to diseases like rust that were present where that population evolved. So it often led to cultivars with single dominant genes for resistance, while the races of the pathogen go through sexual cycles and recombination and selection and develop new races. And so the pathogens would develop new races that would overcome the resistance gene in the variety. And so then the breeders would say, oh, wait a minute, but I got all these other crosses coming along, and wait a minute, there's a cross over here that's got another resistance gene. And so they would go to a new variety with a different resistance gene. And a few years later, the pathogen would say, well, okay, I've got a new race that'll overcome your new resistance gene. And so when we talk about breeding for disease resistance, there's sort of two ways you can overcome this. Probably the more logical way is there also genes that control disease resistance that aren't really single dominant, very strong genes. You can produce quantitative systems that control or actually slow disease development, which are more stable and called horizontal resistance versus these dominant genes vertical. But at any rate, with all of these vertical genes were developed and deployed and broken down, breeders came up with the concept, well, look, we can fix that. We can take one variety, A, and we can add resistance gene 1, 2, 3, 4, 5. And so we have 5 different backcross populations, each with a different resistance gene. And so then if we mix all of those populations together, they all are variety A. We can't distinguish them phenotypically. We mix them together and we get a multiline that will reduce the risk of epidemics, because it's relatively easy for a pathogen population to overcome one of these genes at a time. It's a lot more difficult to overcome 2 and almost impossible to overcome 3 at a time. So that's the concept of multilines. All right, and made it through with 1 minute to spare. Are there questions? Well, yeah, single seed descent would give you problems if you have a crop with inbreeding depression. We'll talk about, Monday we'll talk about breeding cross-pollinated crops as sort of the general principles of cross-pollinated populations and developing open-pollinated varieties. Then Wednesday we'll talk about inbreeding depression. In most cross-pollinated crops, you know, selfing is either undesirable or deadly. And so a single seed descent with a crop like cassava, I mean, if you self one generation of cassava, you're not going to get many seed to descend. And after 2 generations, you're not going to have anything left. So you're not going to do single seed descent with most of our cross-pollinated crops. There are some that are fairly intolerant. I mean, onion doesn't mind inbreeding much at all. And crops like tomato will either inbreed or out, you know, those kind of crops you could do, but for most cross-pollinated, we don't use this. This is primarily used for self-pollinated crops.
After you have the lines, if you cross them, wouldn't that have an effect, sort of like hybridization?
Oh yeah, we'll talk about that. Yeah, hybridization is a sort of the antidote to inbreeding depression is heterosis or hybrid vigor. And cross-pollinated crops, we got the double whammy because you've got this inbreeding depression that gets in your way producing homozygous lines that you can maintain as pure lines to make the same hybrid year after year. But then you get this advantage of hybrid vigor. In self-pollinated crops, we don't have the inbreeding depression to worry about. If there ever was inbreeding depression in self-pollinated crops, it was eliminated because when you get to homozygous loci and homozygous recessive loci that are undesirable, they're tossed out. They don't survive in the population. So with self-pollinated crops, though, the concept of heterosis or hybrid vigor is still very valid, very strong. And so crops like sorghum, rice, and even wheat form very good hybrids. The problem you have with hybrids in some of those crops is how do you produce them economically? How can you get seed production up and functional? All right, so Monday we will talk about a lecture on cross-pollinated, and actually all of next week we'll talk primarily about cross-pollinated crops. Wednesday, inbreeding depression and heterosis, and